We'll be talking about self-assembling peptide nanoparticles and how we use them for biomedical applications. First, I will explain you how we design those particles, and then I will go into the applications. So this here is a small virus. It has icosahedral symmetry. If you look at that, basically you have two-fold, three-fold, and five-fold symmetry elements here. And we were wondering, can we mimic virus-like particles, as you can see here, with just artificial proteins? So our background is basically from protein design. We were working with coiled coils. Here you can see, I can take so these are the axes of the icosahedron. You see the three-fold, the two-fold, and the five-fold symmetry axis. And then if you take coiled coil proteins, like this green helix here, this forms a pentameric coiled coil, and the blue helix here, it forms the trimeric coiled coil. You align it along the three-fold and the five-fold symmetry axis, and you combine them into one single molecule, so that's one single peptide chain, and then you will get something which looks like that. So here you have the trimeric coiled coil, here you have the pentameric coiled coil. They are aligned along this symmetry axis of the icosahedron, and then in a self-assembly process, it will generate a nanoparticle which looks like that. It has icosahedral symmetry, and actually this is nicely visible here. The colors are not exactly matching, but you can see the pentamer here in blue colors and the trimers here in yellow-red colors, and nicely fitting the icosahedral shape of the particle. Looking at not a computer model, on a computer you can do pretty much anything, but looking in reality, this is an EM picture of such nanoparticles, and you can see that they really form very nice homogeneous nanoparticles. So we were thinking about different applications for biomedical applications. Largely, we are looking now into vaccine design. And to design a vaccine, we need to address the immune response. So there are three major types of immune response, and all of them we can address with our nanoparticles. The first one is the B-cell immune response. You get a very strong B-cell immune response when you repetitively display something on a nanoparticle surface. And basically, this means something looks like a virus, and a virus is an infectious agent. So if something looks like a virus, presumably it should also induce a strong immune response. So we take our nanoparticles, we can easily make a kind of Lego with molecular biology. We can engineer different sizes of the particles depending on the size of the protein. So in green you have the pentamer, in blue the trimer, and then in red we stick an epitope from an antigen. See, here this is malaria epitope just on top of the protein chain. So it's still only one single protein chain, and just by making the trimer a little bit longer, here you can see the trimer here is a little bit longer than it is here, you just increase the size of the particles. And if you have a particle of this size, it's a very good immunogen. So we can use that then as a vaccination toolkit. You can engineer pretty much a vaccine against any disease that you're interested in. The second immune response that is important is the T-cell immune response. You can also use the nanoparticles to stimulate the very good T-cell immune response. So how do we do that? We engineer T-cell epitopes into the core of the particles. So T-cell epitopes, it's basically this small sequence here in magenta. That's called PADRA, whatever that is. When it binds to MHC class 2 molecule, like here, so you see the magenta peptide binds to this MHC2 molecules, that triggers then a very strong T-cell immune response. Now, how can we engineer an extended peptide into a coiled coil? Basically, we align the sequence of the peptide of the T cell epitope along with the coil-coil sequence, which has a 7 repeat pattern in which A and D positions are always hydrophobic residues. So if you glide it along that heptide repeat, you might be able to find a pattern that allows you to incorporate the T cell epitope into the core and make it helical. So that's the immune response. On the left side, you see without T cell help. On the right-hand side, with T cell help. Actually, this is an ELISA, and you can see it's a huge improvement of the immune response. The third one of the immune responses that we want to address is the innate immune response. How can you do that? There are some molecules called PAMPs. One of them is flagellin. Flagellin stimulates the innate immune response. This is the flagellin molecule. You can see, since it's a protein, we can simply attach it to the peptide chain forming the particle and then make a co-assembly of a peptide chain that has the B cell epitope, here in red decorated, and then incorporate just one flagellin molecule in the particle. It forms beautiful particles. It stimulates the TLR5 receptor quite a bit, actually even stronger than a native flagellin itself. So when you engineer flagellin onto our nanoparticles, flagellin is even more potent than in its native form. This is the improvement that we get. We get a 200 times stronger immune response from our particles than from KLH, and KLH is one of the standard vaccine carriers that is being used currently. So now to the biomedical applications. One is a universal flu vaccine. So the B cell immune response, we use M2E as a tetramer, helix C as a trimer. The T cell epitope will be padre, and an innate immune response will be triggered by flagellin. How does it look like? So here we have basically, now we have a little bit of a different concept. We combine a trimer with a tetramer helix, because M2E is a tetrameric protein. And if we combine it with a tetramer, we can display M2E in its native conformation. Helix C, which is a trimer, we can display on the trimeric coil, and then we get the nanoparticle, which looks like that. This has now octahedral symmetry, and here you see the flagellin molecule that is also incorporated. This is a biophysical analysis. You have